Researchers at Harvard and the ETH Zurich predict and validate genomic safe harbors for therapeutic genes, enabling safer, more efficient, and predictable gene and cell therapies. Many future gene and cell therapies to treat diseases like cancer, rare genetic, and other conditions could be enhanced in their efficacy, persistence, and predictability by so-called genomic safe harbors (GSHs). These are landing sites in the human genome able to safely accommodate new therapeutic genes without causing other unintended changes in a cell's genome that could pose a risk to patients. However, finding GSHs with potential for clinical translation has been as difficult as finding a lunar landing site for a spacecraft, which has to be in smooth and approachable territory, not too steep and surrounded by large hills or cliffs, provide good visibility and enable a safe return. A GSH, similarly, needs to be accessible by genome editing technologies, free of physical obstacles like genes and other functional sequences, and allow high, stable, and safe expression of a landed therapeutic gene. Thus far, only few candidate GSHs have been explored, and they all come with certain caveats. Either they are located in genomic regions that are relatively dense with genes, which means that one or several of them could be compromised in their function by a therapeutic gene inserted in their vicinity, or they contain genes with roles in cancer development that could be inadvertently activated. In addition, candidate GSHs have not been analyzed for the presence of regulatory elements that, although not being genes themselves, can regulate the expression of genes from afar, nor whether inserted genes change global gene expression patterns in cells across the entire genome. Now, a collaboration of researchers at Harvard's Wyss Institute for Biologically Inspired Engineering, Harvard Medical School, and the ETH Zurich in Switzerland has developed a computational approach to identify GSH sites with significantly higher potential for the safe insertion of therapeutic genes and their durable expression across many cell types. For 2 out of 2000 predicted GSH sites, the team provided an in-depth validation with adaptive T-cell therapies and in vivo gene therapies for skin diseases in mind. By engineering the identified GSH sites to carry a reported gene in T cells and a therapeutic gene in skin cells, respectively, they demonstrated safe and long-lasting expression of the newly introduced genes. The study is published in the journal Cell Reports Methods. While GSHs could be utilized as universal landing platforms for gene targeting, and thus expedite the clinical development of gene and cell therapies, so far no site of the human genome has been fully validated, and all of them are only acceptable for research applications. The researcher says that this makes the collaborative approach that they took toward highly validated GSHs an important step forward. Together with more effective targeted gene integration tools that they develop in the lab, these GSHs could empower a variety of future clinical translation efforts. The researchers first set up a computational pipeline that allowed them to predict regions in the genome with potential for use as GSHs by harnessing the wealth of available sequencing data from human cell lines and tissues. In this step-by-step whole genome scan, the researchers computationally excluded regions encoding proteins, including proteins that have been involved in the formation of tumors, and regions encoding certain types of RNAs with functions in gene expression and other cellular processes. They also eliminated regions that contain so-called enhancer elements, which activate the expression of genes, often from afar, and regions that comprise the centers and ends of chromosomes to avoid mistakes in the replication and segregation of chromosomes during cell division. This left them with around 2000 candidate loci, all to be further investigated for clinical and biotechnological purposes. Out of the 2000 identified GSH sites, the team randomly selected 5 and investigated them in common human cell lines by inserting reporter genes into each of them using a rapid and efficient CRISPR-Cas9 based genome editing strategy. Two of the GSH sites allowed particularly high expression of the inserted reported gene. In fact, significantly higher than expression levels achieved by the team with the same reported gene engineered into two earlier generation GSHs. Importantly, the reporter genes harbored by the two GSH sites did not upregulate any cancer-related genes. This also can become possible because regions in the genome distant from one another in the linear DNA sequence of chromosomes, but near in the three-dimensional genome, in which different regions of folded chromosomes touch each other, can become jointly affected when an additional gene is inserted. To evaluate the two most compelling GSH sites in human cell types with interest for cell and gene therapies, the team investigated them in immune T cells and skin cells respectively. T cells are used in a number of adaptive cell therapies for the treatment of cancer and autoimmune diseases that could be safer if the receptor encoding gene was stably inserted into a GSH. Also, skin diseases caused by harmful mutations in genes controlling the function of cells in different skin layers could potentially be cured by insertion and long-term expression of a healthy copy of the mutated gene into a GSH of dividing skin cells that replenish those layers. The researchers introduced a fluorescent reporter gene into two new GSHs in primary human T cells obtained from blood and a fully functional LAMP3 gene, an extracellular protein in the skin, into the same GSHs in primary human dermal fibroblasts and observed long-lasting activity.